This is a short introduction to the study by Radosevic and colleagues, which was conducted at the University of Copenhagen. In this study, we address the structure of the neuronal network involved in generation of simple, stereotypical rhythmic movements such as breathing, scratching, and locomotion. Although such movement can be quite complex, as we can see in this example with a running cat, the core elements are simple. They consist of rhythm generation and then an orchestration and coordination of different muscle groups. The spinal circuitry behind these movements is not well understood, but models have been proposed. Here are a couple of models that have been proposed for pattern and rhythm generation in the spinal cord. They are quite complicated, but they all share basically the same feed-forward connected structure. So they consist of a interacting part, and then they have a feed-forward part. This means that we can tease apart the circuit into a modular structure. There's a module A that consists primarily of motor neurons and local last-order interneurons. And then there's a module B that is involved in the generation and patterning of the motor activity. We don't know much about module B, neither the size of the neuronal population that it consists of, nor do we know the internal connectivity. Specifically, how are the two modules connected? The nature of the connectivity from B to A all depends on whether module B is large or small compared to module A. There are two possibilities. One is that module B is small compared with module A. For every neuron in module A to receive an appropriate number of synaptic input from B, there has to be a divergent connectivity. This means that neurons in B has many connections in order to drive neurons in A, and for that reason it is also a dense connectivity. The other possibility is that module B is large compared with A. For every neuron in A to receive the same total number of synaptic input from a much larger population in B, the connectivity has to be convergent. It also means that neurons in B reach a much smaller number of neurons in A, and therefore it's a sparse connectivity. So far so good, but how do we distinguish between these two situations? Did you notice that in the divergent scheme there is a large overlap in input among different neurons in A? This will manifest as a strong correlation in synaptic potential across pairs of neurons. The opposite is the case in the convergent scheme, where there will be weak or no correlation in synaptic input among pairs of neurons. This gives us an experimental means to test which of these network structures that exist in spinal motor circuits. Now there is a complication. As I mentioned earlier, motor activity entails coordination of different muscle groups which are not active at the same time. This is believed to be achieved by having multiple modules connected in parallel, but active at different phases. Hence, the slow rhythmic activity will reveal which parallel module a given neuron belongs to. This means that even if the modules are intertwined, we can disentangle the neurons by means of the phase of the slow activity. So we recorded from many pairs of neurons in close proximity of each other and used the correlation in slow activity to quantify their functional relatedness, and the fast correlation to quantify the degree of common source synaptic input. When plotting these against each other, we would expect that pairs that have strong slow correlation belong to the same module and therefore receive common source synaptic input if there is a dense divergent connectivity. This would result in a simple relationship like this. However, this was not what we found. The points scatter around the x-axis, indicating a decoupling between the slow and the fast timescales. In conclusion, we have found a remarkable and surprising lack of correlation in synaptic input between pairs of neurons, in particular the pairs that are functionally related. This indicates that The driving input to motor neurons as well as local interneurons does not originate from the expected small nucleus via divergent connections, but rather from a large network with sparse convergent projections. This is crucial information for understanding the rhythm and pattern generating circuits in the spinal cord, as well as for developing new therapeutic strategies for spinal cord injuries. In discussion, there are alternative explanations for the lack of correlation that we see. One is a phenomenon known as active decorrelation. Active decorrelation has been observed, for instance, in the neocortex, where otherwise synchronized sensory input are desynchronized by local recurrent inhibition. If active desynchronization is found in spinal circuits, it would suggest a novel role for inhibition in these circuits. If you want to learn more, please go to our website, berglab.net, where you can find updates and a link to the paper. Thank you for watching.